In this paper, we formally describe two species of hammerhead flatworms. One species was collected from France and Italy, and the other is from the French island of Mayotte in the Indian Ocean.
The discovery of this new species of flatworm that we discovered was kind of a casual discovery, as many of the other invasive species that we found in Europe. The funny thing was that I received an email from a person, a private person, that simply told me, well, I have this kind of unusual, freaky animal that is appearing every morning in front of my house. Could you please come and check for what it is? So I just replied to the call. I was very excited about, because I wasn't expecting something special, really. But by the time I went there, I mean, I got a very unusual and great discovery. I arrived in this house that is close to the countryside in the Treviso province in the early morning. It was a very foggy and cloudy and very cold morning. And at the beginning, nothing was happening. But as soon as the sun started eating a bit the surface of the house, you could see thousands of flatworms coming out from the garden and then sliming up to the house and to the house entrance. And that was totally unexpected. I mean, something that I've never seen before. And the first thing that I thought was immediately start picking up specimens. And I was able to collect both live and dead specimens. And I suddenly sent all this material to Jean-Loup that as soon as he got the material, he said, wow, this is something never seen before. We definitely need to describe it.
I am J.L.JUSTINE. I am basically a parasitologist with a long career, beginning in 1978. And I am mainly a specialist of parasites of fish. I am a member of the National Museum of Natural History in Paris since 1985, and a professor since 1995. In 2013, a colleague forwarded to me a photograph of a very strange animal found in a garden. And I realized that there was an invasion of these animals progressing in France and Europe, and that nobody was working on them. That was not my field, but I decided to consecrate some of my time to this subject. I sought for international collaborations, and we ended up making astonishing discoveries on these land flatworms. So far, we have published 6 long papers about these land flatworms, all of them in PeerJ. There are 2 main parts in this research. First, a traditional part of taxonomy and description of new species. This was made on specimens we collected since 2013, and most of the work was done by LEIGH Winsor in Australia. He is one of the few scientists in the world who can describe the internal anatomy of these animals.
I'm an adjunct zoologist in the College of Science and Engineering at James Cook University in North Queensland. In Australia, for the last 40 odd years, I've been working on land planarians, mainly Australian and New Zealand species, looking at their biogeography, systematics, and their taxonomy. But I also have an interest in invasive and alien species in countries overseas, because quite often they'll be Australian or New Zealand species that have hitched a ride. Land planarians have few external morphological features that are reliable. This is not a real one, but an example that it's a smooth, shiny species that moves around. There's not much to see apart from the colour, position of body apertures, and perhaps eyes, and the shape of its head. So that these aren't necessarily reliable characters. And ideally, we study mature species by histological methods, looking at them anatomically. The big problem with them is that their organs are embedded in parenchymatous tissue, and it's difficult to dissect this. So you must prepare them in such a way to cut thin sections, such as these ones here, numerous sections right through the worm, and then you reconstruct the anatomy from serial tracings. There's two new species, one of which has been sectioned in that way. The other one, there was insufficient material to do that. So we've relied entirely on molecular evidence for that as a new species. One of the problems that we do have is, and this is something that's quite common in alien and invasive species, is that they're immature. They're not mature in the country where they've been found, such as Europe. And this is where molecular methods are very good. You can either do the DNA barcoding or the mitogenomic work that we've done.
Secondly, there is a modern part about mitochondrial genomes. This was done by ROMAIN Gastineau in Poland. What I like in this research is that we combine the results of field expeditions by Laurent Charles and ENRICO Ruzzier, traditional techniques by LEIGH Winsor, and molecular techniques by Justine J and ROMAIN Gastineau. Real teamwork.
Well, my name is ROMAIN Gastineau, so I'm a French scientist working in Poland right now. My background actually is a PhD in biology of organism, and it was really focused on a very narrow group of diatoms as microalgae. But then I expanded my skills to genomic and bioinformatics, which is how I ended up working with Professor Justine on this flatworm topic. Actually, what we used are next generation sequencing methods instead of just molecular barcoding, as people have been doing for a couple of decades now, which was a very efficient and useful technology. Instead of sequencing single gene, what we tried to do on each and every of the samples we had, and especially the new species we discovered and described, was to sequence complete mitochondrial genomes. In the past, people were using the cytochrome oxidase 1 gene as molecular barcode for molecular taxonomy. Here, and this is mitochondrial gene, of course, and here we sequenced the full chromosome, the full mitochondrial chromosome, using second, mostly generation type of next generation sequencing.
When scientists describe a new species, they have to give a name, which is since Linnaeus in Latin. For the blue species from Mayotte, we chose Mayottensis, which simply means from Mayotte. For the black species from France and Italy, we wanted to do something in relation with the current COVID-19 pandemic. First, most of our research was done when our laboratories were in lockdown. Also, this species has potential to become invasive and spread all over the world. Finally, we were all affected by the pandemic, and we wanted to pay homage to the victims. So we chose the name Covidom. The fact that we describe a new species, Diversi Bipalium mayottensis, from a remote island in the Indian Ocean will surprise nobody. In contrast, describing a new species from Europe is more exceptional. In addition, this species, Humbertium covidum, is an alien species. It is not the first time that an alien hammerhead flatworm is described from specimens in the country they invaded. This was the case for Bipalium QNC, named after the Kew Botanical Gardens in UK, or Bipalium pennsylvanicum, named for Pennsylvania in the US. In both cases, we know that these species originate from Asia. Even worse, there is no confirmed records of Bipalium pennsylvanicum in Asia, meaning that the species are spread over several states in the USA, but has never been collected from its land of origin. The same is true for our new species, Humbertium covidum. We have tried to find mentions of these very characteristic black species in the literature. To date, we have only found a very few possible records in Far East Asia, but no definitive proof that it is the same species. This illustrates the main problem with invasive species. They proliferate in the country they invade because they find abundant food and have no enemies, even when they are rare and discrete in their land of origin.
Well, what I can say, well, formally, I'm first of all a nature lover. I've always been very in biodiversity and nature since I was a child. So I have to confess that I'm one of those lucky people and that have been capable to translate their passion and make it their work. I mean, I'm being very lucky, I would say. During my experience, I had the chance to work in different institutes that helped me to grow up in the scientist that I'm at the moment. And very great experiences, for example, when I was in Spain and the Biological Institute of Evolutionary Biology, the Natural History Museum in London. And since 2010, and now I'm a research fellow at the DAFNI department at Padua University. And what else I can say? I define myself an entomologist, but despite this, I definitely love everything about biodiversity in general. And my specific field of research is focused on beetles, Coleoptera, diversity, functional morphology, and many applied side in applied entomology. I have to confess that recently, well, since I moved in to Padua, I started devoting part of my interest in invasive species. And this is one of the reasons that brought me here, because it's one of the discoveries that I did that allowed us to publish this paper.
Well, submitting a paper to PeerJ is great. At the end, we have a nice message telling you that it's time to have a good coffee, a well-deserved copy, and it works. I believe I convinced already many of my colleagues to publish in PeerJ. In addition, I am also an academic editor for PeerJ because I am a full partisan of open access in science, as long as it is not scandalously expensive. I published my first paper in PeerJ in 2013, and I paid my personal subscription then. And since then, I never had to pay again, except when we publish very, very long papers.
I definitely recommend other people all over Europe, at any time they come across something unusual, to send us an advice, telling us that, okay, we found something strange in our garden. Please, can you check it and let us know?